I'm excited to moderate this microbiology session, and our first speaker is Dr. Raya Faigenbaum Romm from the Hebrew University.
Hi, thanks, Idan. And I'm really excited to present my work. So I want to tell you today about uncovering fitness strategies inherited from single cells with microcolony sec. But first, I want to focus on Staph aureus bloodstream infections, and more specifically on the treatment failure of these life-threatening infections. Recently, promising treatments have failed clinical trials. But why? I want to argue that this is because of bacterial heterogeneity. One treatment was by using a monoclonal antibody targeting the CLFA gene. This is an adhesion factor that Staph aureus bacteria use to attach to the host cells. But Staph aureus has multiple adhesion factors, meaning that other bacteria may express other adhesion factors. Therefore, blocking this gene alone may not effectively prevent bacterial attachment and immune invasion. So we need to understand the coexisting bacterial states within an infection for crucial elimination and effective elimination of the infection. But how can we detect heterogeneity in the bacterial culture? If we use bulk RNA sequencing on the culture, we simply get the average expression of the coexisting subpopulations. Recently, single-cell RNA sequencing techniques have been developed in bacteria. These are powerful techniques to determine the heterogeneity at a specific time point, but they contain several limitations. One is that they don't differentiate between heritable and transient heterogeneity. They also don't distinguish between genetic and phenotypic variability. And also, we're not able to regrow the determined subsets and to study their fitness advantage. So we developed microcolony-seq. Our method focuses on microcolonies formed from the original culture. And more specifically, our method aims to detect single-cell inherited heterogeneity. We want to be able to determine the fitness advantage of each subpopulation and also to differentiate between genetic and phenotypic heterogeneity. So our method starts by simply plating the culture you want to know heterogeneity in on an agar plate. Now we want to determine the time point where very small colonies, microcolonies, are starting to appear. This is very important to sample these colonies at the exponential phase, because we see that some phenotypic heterogeneity actually is lost when the colonies are at stationary phase, like overnight regular colonies that we use. For that, we use the scanlag methodology. This is an automated imaging technique developed in the lab. Therefore, it enables us to determine the exact time point very small colonies, microcolonies, are starting to appear. We simply pick each microcolony and we freeze part of it for further characterization and whole genome sequencing. And we also apply RNA sequencing on the microcolony. So we use bulk RNA sequencing, but not on the entire culture, but on the microcolonies, individual microcolonies formed from the culture. And then we have a computational pipeline that enables us to determine the subpopulations. Our computational pipeline has a step for identifying variable genes. In this plot, in the x-axis is the mean expression of each gene in the bacterial genome. Each dot here is a gene. And the y-axis is the dispersion. This is a measure for variability. In yellow, you can see the genes that we marked as the variable genes. Now, we take these variable genes and we cluster the microcolonies using only the variable genes. You can see here that we determine in this hierarchical clustering two subpopulations of microcolonies. Now that we know that subpopulations, we can apply differential expression analysis, fitness characterization to understand these subpopulations. We applied microcolony-seq on three main human pathogens, on EPEC, enteropathogenic E. coli, under host mimicking conditions. We applied it on UPEC from a urinary tract infection sample. We obtained a urine from the clinic and we simply directly plated it on an agar plate. We identified phenotypic heterogeneity. And we also applied it on Staph aureus gram-positive bacteria from a bloodstream infection, bacteremia. And this part was done in collaboration with Professor Maskit Bar-Meir from Shaarei Tzedek Medical Center. And I want to focus on this part to show our results. So as you know, bloodstream infections have a very low bacterial load. When we plated about 1 ml of blood from the patient with a bloodstream infection on an agar plate, we obtained 23 Staph aureus microcolonies. We applied microcolony sec and we were able to determine 3 main subpopulations, marked here as A, B, C. We also applied whole genome sequencing on each microcolony and we were able to determine that no genetic difference was between these subpopulations, meaning that the heterogeneity in the Staph aureus microcolonies was phenotypic. Now, we were able to apply differential expression analysis to study these subpopulations. When we compared between subpopulation A versus B and C, we saw an elevation in the A subpopulation of capsule genes and other virulence factors of Staph aureus. So we named the A subpopulation as the virulent one, B was in the intermediate state, and C was the avirulent subpopulation. We notice an elevation of arginine and histidine metabolic processes that are linked in Staph aureus to acid response. So we were now able to ask, is this elevation functional? By simply taking the part of the microcolonies that we saved from the beginning of the experiment and growing them at acidic conditions. You can see here that we identified a statistically significant advantage of the Vir subpopulation in acidic conditions over the other 2 subpopulations. When we first grow the 3 subpopulations to stationary phase and then transform it to acidic conditions, the advantage was erased, meaning that there is a reset of the phenotypic heterogeneity at stationary phase, and that this heterogeneity would not be detected in clinical settings because cultures in the clinic are routinely grown to stationary phase. Therefore, this interesting and important phenotypic heterogeneity is erased at clinical settings. But you can ask me, why would bacteria coming from the human blood be in acidic conditions? The human blood is actually slightly basic. But we also know that this specific patient was diagnosed with bacteremia, bloodstream infection, with endocarditis. Endocarditis is an infection in the heart's valve, and it was also found to contain a biofilm. The biofilm of the Staph aureus is slightly acidic, meaning that we can assume that the subpopulation of the virulent subpopulation was actually containing a memory of the biofilm environment, while the other 2 subpopulations, the intermediate and the Vir one, were planktonic bacteria from the blood. But so microcolony sec determines the bacterial state within the host. But you remember that I started to say that to eliminate an infection, we need to understand the coexisting subpopulations, and we also need to understand which drug combinations would eliminate all these subpopulations. For this specific patient, we have these 3 subpopulations. Current treatments that are under development for Staph aureus bacteremia, for example, this treatment that targets capsule genes and this treatment that targets CLFA gene would target only the Vir subpopulation, leaving the 2 other subpopulations in the patient. This antibody that targets the SPA protein would target only the intermediate subpopulation, meaning that the failure of the 3 clinical trials using these treatments was possibly due to the untargeted subpopulations. Microcolony sec can suggest which drug combinations to use to eliminate and to target all coexisting subpopulations. So I showed you that our method identifies single-cell inherited variability. because we look at the microcolony that is uniform and contains the memory of the founding bacterium. We identify with bulk sequencing coverage the expression level of all the genes in the bacterial genome. We are able to differentiate between genetic and phenotypic heterogeneity by applying RNA sequencing and whole genome sequencing on each microcolony. We also identified new virulence genes and new virulence programs. And most importantly, it is very easy to implement. You don't need any special equipment in your lab. So we started when we plated our culture on the agar plate, we obtained very similar looking colonies. But now we know that they are phenotypically different. So I want to thank Professor Natalie Balaban, Professor Ilan Rosenshine, and Professor Maskit Bar Meir, and of course Science Abroad for giving me the opportunity to present my work today. Thank you.
It was great. Thank you so much, Raya. One question from the moderator. Can you please speculate on the mechanism for the reset at stationary phase, and why do you think it exists, if there's benefit to these subpopulations?
Thank you. So specifically for the EPEC, enteropathogenic E. coli, we studied the mechanism of the reset, and we found that a specific virulence factor is highly expressed at exponential phase and is downregulated at stationary phase. And when we overexpress this regulator at stationary phase, we found that there is no reset. So specifically for EPEC, we were able to understand a little bit about the mechanism. For the other pathogens, we're still studying.
Okay, thank you. Any more questions? Okay, so I think thank you, Raya. It's great.
Thank you.
And